Thank you for joining the Once Changing the World, which is India's first future tech meets sustainability podcast. And today, I'm delighted and honored to have with me Professor Thomas Hartung, who is a biochemist and physician with a focus on cellular and molecular research into diseases. He's currently the director of the Center for Alternatives to Animal Testing and a professor of evidence-based toxicology with a joint department in the Department of Molecular Microbiology and Immunology. Dr. Hartung's work spans from pharmacology, toxicology, to molecular microbiology and immunology, with a special focus on the development of cellular methods and their quality control. He has worked on supportive therapies to cancer treatment, inflammatory processes, infectious diseases, and cytokine therapies. Dr. Hartung is interested in the development and quality control of in silico and in vitro tools and their integration into testing strategies. Dr. Hartung's research group has developed a highly standardized human brain organoid, which is currently commercialized via Axosim for a number of viral, neurodevelopmental, neurodegenerative, neurotoxic, and other disease models. He has authored more than 625 scientific publications, which were cited more than 40,000 times. And so today the topic of conversation is obviously going to be organoid intelligence. So Professor, I really appreciate you and taking time and being part of a humble podcast. Why don't we start with maybe explaining what are brain organoids?
Good. So thanks for having me, first of all. Brain organoids are part of a revolution taking place in biomedicine, which was started with the availability of stem cells. Human stem cells became available in 1999, but these were embryonic stem cells. And in 2006, this ethical dilemma was solved by induced pluripotent stem cells. This gave a Nobel Prize after 6 years, unheard of, because it is really promising to make available human tissues in high quality. We call them microphysiological systems, and this is everything which is producing an architecture and a functionality of an organ. And for the brain, this means quite a lot. There's quite a few cell types, various types of neurons, glial cells, and they interact to form networks. The glial cells form support structures like the myelin shield. And these are very important to ultimately achieve some functionalities. And this is what a brain organoid is about.
Could you elaborate on this, Professor? Because of what you're saying, what I understand is that you take a stem cell, and through the stem cell, you create an induced pluripotent stem cell. And through that, you keep it in a petri dish and you feed it whatever proteins and whatever cells. And these kind of develops into different organoids. It could be a brain organoid, it could be a lung, it could be whatever you wish it to be. So could you define or maybe elaborate this process about this creating this brain organoid? When you create this brain organoid, now our brain is extremely complex. It's got 80 billion neurons, 100 trillion synapses. So when you say that you've developing a brain organoid, how similar it is to the brain, structure as well as functional wise?
So the first step is to get these stem cell lines. The starting point is a donor, somebody who gives cells. This can be a healthy person. This can be a patient with a certain disease, for example, a genetic disease, which then will also change later whatever tissue we derive. In the beginning, it was typically a skin cell, so a tiny piece of skin from surgery or just taken as a small donation. Nowadays, we work often with blood cells, or we just take urine, because the few cells which you find in urine are sufficient to produce stem cell lines. So with the consent of the donor, we can start research. And the first step is to reprogram these cells to become stem cells. This is the invention by Yamanaka by adding 3 factors. he showed we can produce something which is very, very close to an embryonic stem cell. And you can do exactly what an embryo does, form all of the different tissues from it. And we have seen since 2006 an enormous number of publications. Every month we are adding to even better models of this organ, that organ, and combining this with all of the tools of bioengineering. In our case, we were in 2016 only the 4th group to produce brain organoids, but we were the first group to mass produce them. So our contribution was really thousands of identical brain organoids, round balls of cells, which, however, replicate some of the architecture of the human brain. So, for example, we found from the beginning about 30% helper cells. So it's really a self-organizational process which is stimulated with some growth factors. So it is a very accelerated development compared to human brain development. We don't have to wait years until you get a functional human brain. We can get something which is at least showing some of the functionalities of a brain after some months. And this is really accomplished by the capability of stem cells to run through a program which is creating these complex structures. And we're only helping, we're only fueling it with cell culture media, oxygen, and some growth factors.
How cool is this? There's nature, which is doing some really, really crazy things. And eventually, when we understand what it's doing and how you just help it develop into things, I wonder what could be possible in the near future. Could we end up creating our own full-fledged brains or full-fledged organs, so rather than waiting for an organ line, for somebody, you could just go and possibly maybe, rather than waiting for somebody you donate, you could get, so very, very interesting. Professor, can you elaborate on this? Because you mentioned you're the only team that's mass-produced these brain organoids. What do you do with these mass-produced brain organoids?
Yeah. In 2016, we were the first. In the meantime, this has become a pretty common technology, and there's even commercial solutions for this available. So you can order brain organoids from some places. The important thing in mass production is that we are producing small balls of cells, which are about 0.5 millimeter, about 50 to 100,000 cells. They're just visible. But the important thing is we have thousands of them, and they have the same composition, not only from one organoid to the other, but also next week and next month. And if it's done in a different lab, it is also quite reproducible. So this is the prerequisite for really testing. So I'm, as a pharmacologist and toxicologist, I'm interested to find substances which are interfering with brain development. I'm interested in autism. Why our children are developing these problems? It is not only genetics. There are certainly chemical components. And we have used over the last years these models to show that certain pesticides, that certain drugs, that flame retardants are interfering with the normal development of the brain. So we can look what is critical, what is suspicious, and what seems to be clean. In a similar way, you can use this model for other diseases. We have been interested in virus infections, and when COVID hit, the first thing I thought was when I heard in February of 2020 that many patients showed neurological symptoms. Could this virus infect the human brain? And it was only in May that we succeeded to show, unfortunately, that human neurons can be infected with this virus. Because we found partners here in Hopkins, and we instantly infected the human brain with clinical strains and saw that there is infection. This has been now confirmed in many other laboratories, and also the clinics is indicating that some patients get a brain infection, which is really bad news because you never get rid of a virus which is in your brain. And we only start learning what this means. other work we have done was on Parkinson's disease. We showed that substances which produce Parkinson in animals, but also in humans, are doing just the same thing in these brain organoids, which is eliminating dopaminergic neurons. Another story was we grafted patient-derived glioblastoma. So patient had a glioblastoma, surgeons did remove it, and we took these cells and added them to our brain organoids. So we could then produce thousands of these tiny little brain organoids in which a small glioblastoma was growing. And then you can try which chemotherapy is working. This is interesting not only for developing new chemotherapies, it is also possibly a way forward to find the exact treatment for a patient. What is most effective? Because we have more than 200 chemotherapeutics available, and you only have a few shots in this devastating disease.
Wow. So what I hear from you, Professor, is that through this, you could model the patient's through the urine, blood samples, or skin cell, and model the patient's cell, a brain organoid with 50 or 100,000 cells, and you could possibly understand what the problem is, and then eventually figure out a way where you could go to precisely maybe help that specific patient with a precision medicine approach. Tell me a little bit more about this. Where does this research stand? Because there are thousands or millions of families around the world who have various neurological condition, from autism to Down syndrome to so many others, and you mentioned about cancer also. When do you think something like this could go from lab to the market? Because there's millions and millions of people who would really, really benefit from this.
I mean, this is already reality. We are already producing brain organoids from children with certain neurodevelopmental diseases, and we are trying out with our partners certain therapies to correct this. We are at this moment trying to expand this also to the neurodegenerative diseases, so Alzheimer's disease, amyotrophic lateral sclerosis, and others. But here it is much more difficult. We first have to show that a young brain can actually show something which only happens in an old person, and it's about aging. One of the big restrictions, however, I have to say, is if I take a specific patient, it takes me almost a year until I have brain organoids. This is a big restriction because if you have a patient, you typically want to intervene very quickly. So in case of the cancer patients I mentioned, we used the simple trick that we used the healthy brain organoids from somebody else and then entered only the cancer system, which is not a limitation. It does not take the reprogramming and finding the best cell line out of these reprogrammed cell lines, which takes us about a year with all quality controls. And for this reason, different diseases pose different challenges, but it is already active research to use them as disease models and to use them really to find treatments.
You're also part of Axosim. Would you like to talk about Axosim?
Happy to do so. I mean, I tried first to create my own company around the brain organoids as a spin-off from Hopkins. We called this Organome, but I failed miserably. I'm not a good businessman. I'm a scientist. And I was very happy that I found partners in Axosim, which is a startup in New Orleans. They have very successful models of the peripheral nervous system. So very similar. They're interested in all the nerve cells outside of the brain. And they have developed models which help now to find out reasons and cures for substances which are hitting on this peripheral nervous system. So it was a very good match that they took over licenses from Hopkins to produce now also the brain organoids. And this is one of their goals. For me, it is a way of having a partner which can offer commercial services, and I can concentrate myself on research and on the future. while others are exploiting the fruits of our results.
Recently, I think you've been in the news because of organoid intelligence. So far, we've had classical computers, which works in binaries, zeros and ones. We are also kind of trying to develop quantum computers, which will work with qubits, sourcing the nature itself, the atoms. Then there is biocomputers. So, can you start explaining what biocomputers are? And how are you leveraging brain organoids intelligence to build the future of this computing?
I mean, brain computers is science fiction. Yeah, this is still far, far away. But the important message at the moment is that we are really moving forward with the basic steps of which is starting to get cognitive functions in a dish, in a cell culture. I said earlier, microphysiological systems that's when we get the architecture and the functionality of an organ produced. In the case of the brain, the functionality is cognition. It is learning, memory, and these things. So we were wondering, how far can we go? Can these systems actually learn? Can we demonstrate these type of functionalities? And we teamed up with a very large team. In the meantime, about 80 researchers are working with us to form what we call the Organoid Intelligence community, OI. Our partners from Cortical Labs, for example, showed in November that brain cell cultures can learn to play Pong, this simple computer game of the 70s. And they're working now with our organoids to find out whether they can also create long-term memory for this. And in very similar ways, we are working with colleagues here in Hopkins in other type of virtual environments, but also to control robots by brain organoids. Similar work is done in other labs. For example, Alison Motri, a collaborator of ours and co-author on the Organoid Intelligence, he is in UC San Diego, and he already controls robots which are walking with a brain organoid in an incubator, and through Wi-Fi, they're communicating, and he claims that the robot starts walking and exploring its environment. So there's a lot of fascinating things happening. At the moment, a lot of them are anecdotal or just, we achieved something, but we want to really form a community which is systematically looking into this. And this is why we held a workshop last year, which was published last week on the 28th of February. And we are, and this workshop report and the vision paper we published at the same time is trying to tell the scientific community there's a synergy now of different technologies which allow us to study cognitive functions in cell culture. And this opens the gates for really a lot of applications. It is really the synergy of biomedicine, producing these stem cell-derived models of a brain organoid, electrophysiology as a sensor technology, which is advancing very well, and artificial intelligence, computational approaches to make sense of the signals and reading them. And we see that this is really creating three major possibilities. The first one is just to study how the brain works. Because so far, if you want to know how a brain works, you're very limited what you can do in humans. Animals have always the problem of ethics, but also that they're not humans. They're not thinking like us, And it is very expensive to work on animals too. So we solve some problems by making this available. The second piece is if you have such a model, you can look into substances which might impair this. Nobody knows whether Alzheimer, for example, is also fueled by some toxic chemicals. We see that the numbers are increasing, but they're increasing more than we age. It's not just that we have more older people. It is also something which is getting more and more frequent. Autism is another example. Autism was seen in 1 in 10,000 children in the US in the 70s, and now we have it in 1 in 44. This was the last data from the CDC. So we would love to see whether chemicals contribute to this. And we believe that cognition would be a very sensitive endpoint, because that's where the problem is. Yeah, whether my memory does work properly, whether I can learn about my environment properly. So this would possibly enable, also by producing the brain organoids from patients with a certain genetic background, to really find models, to find new drugs, to make it better, to control, to correct the deficits. And then the third aim, which is certainly the long-term aim, is to bring this to computation, a biological computer. I would say we are certainly far away from something which even matches what a computer can do. A simple computer, yeah. But you know how these technologies are. They're synergizing, and sometimes you see miracles happening, how fast things can develop. And we might also, just by learning how the brain works, optimize our computers to make them better. Because there's still an enormous difference between a human brain and a computer. To give you a few examples, the best computer in the world, a supercomputer, the Frontier computer in Kentucky, matched the computational capacity of one human brain only in June of last year. The brain is calculated or estimated at 1 exaflop computational power. This is exactly what was reached by the Frontier computer in June of 2022. We also work differently than a computer. We have intuition. We can take decisions on incomplete data, which a computer can't. And this has a lot of consequences. So, for example, if you want to train a child on distinguishing cats and dogs, you have to show them about 10 pictures, and then they are pretty good in distinguishing cats and dogs. A computer needs hundreds, if not thousands, to do the same, to get the same error rate. Or let's say you want to learn a new language, and you add some new vocabulary. A computer has to rerun the entire program. We simply add them. We call this progressive learning. So these are all capabilities we really would like to see in our computers. Also, the efficiency. If you compare again to this supercomputer, the computer used 1 million times more energy than our human brain to achieve this computation. And it is a $600 million installation on 680 square meters. So it's really a big thing, yeah. But we have in just 1.4 kilogram of our brain.
I wonder what the world will look like when we transition from a traditional computing paradigm to possibly biocomputing, maybe where we have something in the lines of maybe you're leveraging, organoid intelligence, what you're working at. So you pointed out obviously at the wrongs. I mean, the problems with or traditional computing, and yes, I mean there's no debating that our human brain is extremely fascinating, and the way it computes, even supercomputers are not able to match up at this point in time. What could be the challenges of building a biocomputer, organoid intelligence that you're talking about? And you did give a glimpse of what could be those applications, but could you maybe elaborate on that? And when we actually, what are the timelines first you see of possibly working towards an organoid intelligence, and maybe elaborate on possibly the applications and how the world would look like. So then there's more people joining in this effort to kind of drive this innovation forward.
I mean, the first thing is you need many disciplines to work together here. And this includes also ethicists, yeah, because you really have to be very careful what you're doing. But it starts with really neurobiologists, then bioengineers, then electroengineers, data science specialists, and then brain-machine interfaces, and all of these. Yeah, it's a big challenge, but I think the possible profits are enormous. But this requires us also to find first the right vocabulary. So we are at the moment working on defining what is the right term. Should we call about intelligence in a dish, or what is intelligence? Yeah, what is intelligence? Intelligent behavior of a cell. Doing 1 + 1 is 2 is not intelligence. This is a trained response. But if I only learn 1 + 1 is 2, and then on my own find out that 1 + 1 + 1 is 3, then it is intelligent. So transfer learning is, in this sense, is an important property. And so we have really defining the minimums at the moment while trying to build up examples which attract funding to make this research happen. Because, I mean, this is not a simple thing. It requires really substantial amounts of investment to move forward. But I think that we have already good examples that in principle, these biological systems are capable of some basic elements of cognitive functions. But now we have to standardize them. We need benchmarks. You can only say this brain is working better than that brain if you have something to measure it. This is the elegance of this Pong playing approach, because you can simply ask how often did the brain fail to get the ball bouncing back. Yeah, you can start to set a benchmark, and then you add a substance and see whether it becomes better in the next training session. So these are the type of things which are coming into reach, and I believe that it only takes us a few years to really have models of basic elements of learning. And you will be surprised as soon as you look into the literature how many of these things are not clear. We don't really know how information is stored in the connections of these brains, because it's the connectome. You referred already to the 80 or 100 billion neurons. A neuron has about 1,000 connections to other neurons. So this is how we store a lot of the information, very different to our transistors and chips, and we permanently build connections and take them away. And this is how we store memory. But having an experimental system to analyze this is a tremendous opportunity to really get the machinery cleared.
Right. You mentioned about ethics, and we believe that intelligence is emergent. With these organoid intelligence, do you see these organoids intelligence becoming conscious like these brain organoids? Is there something? Is that a possibility?
This is certainly something which is far away from what we are doing at the moment. You have to imagine the brain organoids, which we are typically working with at the moment, have about 50,000 to 100,000 cells. That's about what the nervous system of a fly. So this is already limiting what we can do. But we have to say we are moving forward. We are increasing this, which has some technological challenges, because you need to find ways to get oxygen and nutrients into the center of these organoids, something which is equivalent to blood system, yeah, so blood vessels. But the main thing for consciousness is you need to develop experiences. So it is about the inputs and then the feedback to my outputs, which makes this happen. So, for example, one of the questions always posed is, can these systems suffer? They can certainly not have pain, because there's no pain receptor in the system. There's nothing which signals, oh, there's a painful experience. And in a very similar sense, as long as we are feeding them zeros and ones, computational inputs, we will not see any consciousness developing. But this might change at some point. If we are able to combine it, let's say, with a retina organoid or an artificial eye, there might be other experiences which are coming in. But can you develop consciousness without emotions, without empathy, without all of these elements? So by what you feed in, you also control very much on what the brain can do. I'm not a philosopher, I'm not an ethicist, but I can tell you that the ethicists we are working with are very excited by the idea to define where does it start? When can you say a system is sentient? It is self-aware of its environment. So what would be a test to establish this? This is a very interesting question. And then also the question comes at what would the general public feel about it? So we are serving at the moment, or my colleagues in the Berman Institute of Bioethics, they are really serving people and ask, when does it start to get uneasy? And then they give them information like, yeah, this is done with the informed consent by the donor of the cells, or this is done to find drugs for Alzheimer. How does it change the attitude? Because we want from the beginning to communicate this properly, that people say, yeah, what you're doing is acceptable, is for good cause, and it is done with the limits society should set for any type of research which is bordering to ethical problems. Yeah.
Right. Professor, thank you. Really appreciate you taking time and being part of the podcast. I think you are possibly building something which could completely upend the world. I think the entire healthcare industry is going to get upended. The world is going to possibly ask maybe more deeper question in what is intelligence, what is God. I think all these questions will completely get upended. You spoke in the middle about your partner's cortical labs using Organoid Intelligence for playing Pong games. You spoke about Alison Moultrie and his team leveraging the same thing for controlling robots. Maybe end the conversation with what is it that you personally are most excited for? What is it, that one single goal that you are trying to achieve with Organoid Intelligence?
Actually, I'm from the beginning you heard I'm very broad. I'm interested in many things. I'm interested in creating partnerships. I'm interested in getting things moving, and I see how our humble starting points at the moment create enthusiasm, possibly create really a part of science which did not exist before. And I see the immediate impact for what is close to my heart, which is treating diseases, understanding and treating diseases. I'm very excited that I was part of the start, and our humble contributions at the moment are inspiring others to carry this forward. Yeah.
Professor, really appreciate, truly honored and a privilege to be speaking to you. Wish you and the team the very best, and I hope that what you're building comes to fruition very, very soon. It moves from lab to the market and touches people and changes lives and changes healthcare, and we get into the world of precision regenerative medicine. Really appreciate you being part of the podcast. And to my listeners, if you like what you see in here, then please press the subscribe button. And until next time, see you guys. Bye-bye. Thank you. Thank you, Professor. Really appreciate this.
Thanks for having me.